Hello everyone. Today we are going to start a quick review about lac operon. This is a lac operon consisting of polycistronic gene. Polycistronic gene, poly means many and cistronic gene. So it is made up of I gene, Z gene, Y gene, A gene. I gene will code for a repressor protein. This is a protein which is a product of I gene and it will form repressor protein through mRNA. This this piece of DNA will form mRNA, mRNA will form protein. In the absence of inducer, when what is inducer? Inducer is lactose or allolactose. In the absence of inducer, when lactose is not present, this repressor protein will bind to the operator region and prevent RNA polymerase. This is RNA polymerase which will work in this side in the presence of lactose. It will bind with RNA polymerase and prevents its polymerization, and the product of Z gene, Y gene and A gene will not be formed. Will not be formed. But in the presence of inducer, when lactose is present, this Z gene will form beta galactosidase, and beta galactosidase convert lactose into glucose and galactose. And this permease will the bacteria help the bacteria to absorb lactose. And the product permease is a product of Y gene, and the product of A gene will be trans-acetylase, whose function is not known. They also will form through mRNA. A piece of mRNA will be formed, which will form enzymes. In the presence of inducer such as lactose, in the presence of inducer, when inducer is present, lactose is present, it will bind to the inducer protein. It will bind to the repressor protein here, and now this repressor protein will not able to combine with O protein, and this RNA polymerase will be free to work in this direction. And this RNA polymerase, what will do, it will form mRNA from the piece of DNA. It will form RNA from DNA, RNA from DNA, and this RNA will form beta galactosidase, primase, and trans-acetylase. Lac-operon was first demonstrated by Francois Jacob and Jacques Monod. Lac-operon is polycistronic structural gene, that is, it is made up of many genes. It consists of one regulator gene and three structural gene, total 4 genes. The I gene code for regulator gene, Z gene code for beta galactosidase or beta gal, and Y gene codes for permease, which increases the permeability of galactose, and A gene codes for trans-acetylase. The function of beta galactosidase is fermentation of lactose into glucose and galactose. The function of permease is to increase the permeability of beta galactoside. Beta galactoside is a lactose, and beta galactosidase, which is ACE, is enzyme which will digest lactose. So beta galactoside is a lactose and beta galactosidase is an enzyme. On and off of lac-operon is lactose and allolactose. This is important. Allolactose is also on and off of lac-operon. Inducer of lac-operon is lactose. Does the lac-operon also operate in the absence of lactose? It is yes, because in the absence of lactose also the product of lac-operon is formed in a small amount, and especially permease. which helps the bacteria to absorb lactose in initial stage. Which protein is all the time in the absence of lactose? It is a repressor protein which is formed in the absence of lactose, and it will bind to the operator region. The function of a repressor protein is to bind with the operator region and prevent RNA polymerase to form the mRNA of Z, Y, A and A gene. The lac operon can also be called as a regulation of enzyme in the presence of substrate. Substrate is lactose. So in the presence of lactose, enzyme is formed. In absence of lactose, enzyme is not formed. The regulation of lac operon by a repressor is termed as negative regulation because a repressor protein inhibits the RNA polymerization of DNA. For latest update, please subscribe and do not forget to comment and share. Thank you.